Today we're going to discuss population genetics and allele frequencies and how they interact with equilibrium in populations that we can calculate through something called Hardy Weinberg equilibrium. So first of all, what is population genetics? Population genetics is simply the study of the rules governing the maintenance and transmission of genetic variation in natural populations. So there's a lot to unpack here, and in the coming slides, many of these terms will be made clearer to you. But the biggest question is, why does this matter? Why are we talking about population genetics? Well, if you remember from our introduction to evolution, evolution happens at the population level. And population genetics directly intersects with evolution in what we call the modern synthesis. And the modern synthesis describes how evolutionary processes, like natural selection, can affect a population's genetic makeup, and in turn, how this can result in gradual evolution of a population and speciation. So the modern synthesis represents the combination of genetics and evolution. The modern synthesis provides a coherent understanding of the relationship between natural selection and genetics, and it allows us to connect population change over time, which is what we refer to as microevolution, to the processes that gave rise to new species and higher taxonomic groups with widely divergent characteristics and traits. We call that macroevolution. So the modern synthesis allows us to connect micro and macro evolution. In the definition of population genetics, there was a mention of rules. So what are these rules founded on? The rules for population genetics are founded on the idea of differences in allele frequencies. Allele frequencies mean the rate at which a specific allele or a gene variant, a genetic variant, appears within a population. These allele frequencies or genetic variants have variability in their genotype, so the gene makeup, and also, as a direct result of the genes, a variation in phenotype, which is the observable characteristics that result when the genotype interacts with the environment. And we can put all of this in a cartoon that represents what we are talking about. Here we have the entire gene pool, which is affected by sexual recombination, mutation events, as well as gene flow by way of migration into the population. And all of this leads to population-environment interactions that involve natural selection processes, genetic drift, and ultimately evolution. Now we'll talk about what all of these terms mean in detail in a minute, but first we need to talk about genetics for a second. This is Biology 113, not 115, so we're not going to talk about nitty-gritty details, but it is good to have a basic understanding of the foundation of the concepts that we're talking about. So we had a, quote, rediscovery of Mendel's genetic studies in 1902 by someone named William Bateson. They completed the missing model for inheritance of genetic factors by looking over books and research written by Gregor Mendel. And Gregor Mendel published his work in the Transaction of the Brünn Society of Natural History in 1866. What he did is he planted pea plants in the garden. He was a monk and his passion was gardening. And through this passion, he was able to explore different genetic variants of the pea plants that he was growing. And through his studies, he established two set laws or rules that came up time and time again in his work. Mendel's laws include number one, the law of segregation. This law states that paired unit factors or genes must segregate equally into gametes such that the offspring have an equal likelihood of inheriting either factor. And number two, independent assortment. This is Mendel's law of independent assortment, stating that genes do not influence each other with regard to the sorting of alleles into gametes, and every possible combination of alleles for every gene is equally likely to occur. There's a lot to unpack in both of these laws. There are some words and terms we may not be completely familiar with. So let's go through an example here when we are talking about pea plants specifically. So here we have two peas, one that has a smooth phenotype and one that has a wrinkled phenotype. That is their physical appearance. This is our parental generation. Two homozygous peas each have two copies of the same allele. What we mean by allele is the gene variant for either smooth or wrinkled phenotype. Big R, capital R, is the allele for a smooth phenotype. Little r is the allele for the wrinkled phenotype. So the genotype for smooth here is homozygous, and we know that the dominant allele is capital R, representing the smooth phenotype. So this parent has two copies of the dominant allele, big R, big R. By contrast, the wrinkled parent has two copies of the recessive allele for the wrinkled phenotype, little r, little r. So that is what is meant by homozygous. The two alleles that each parent has are both the same. The smooth parent has two big R's for the dominant allele, and the wrinkled parent has two little r's for the recessive allele. These then produce reproductive cells, which are called gametes. Each gamete produced by the pea plant carries one allele. All gametes from the double recessive plant or the homozygous recessive plant carry the little r allele. All gametes from the big R plant carry only big R because it is homozygous dominant. So this is the representation of the two sets of gametes that are produced by the parents. We have only big R's here and only little r's here. All dominant, all recessive. In the first generation, we can see what offspring are produced. Every offspring from the two parents inherit a big R, a dominant allele from the smooth parent, and a little r, a recessive allele from the wrinkled parent. Because big R is dominant, all the new peas are smooth. Here are their offspring. The only possibilities that could have arisen, taking one allele from parent 1 and one allele from parent 2, is the condition of heterozygous dominant. It's called heterozygous because it has both the dominant allele, big R, and the recessive allele, little r. And because the trait of smoothness is dominant here, all of the offspring in the F1 generation are smooth. They have a smooth phenotype. We can then do another cross between this generation and take a look at F2, our second generation. This is where things get a little tricky and we have to do Punnett squares. In the F2 offspring, each gamete produced by the heterozygous parent has a 50% probability of being big R and a 50% probability of being little r. As a result, they produce equal numbers of big R and little r gametes. This is how we set up the Punnett square. Over here, we take parent 1 and stick it on the left axis. We see that we have both big R and little r. So we put the big R here, the little r here. And then the top axis, we put the second parent. We take the big R and put it here, the little r and put it here. So those are all of the possible gametes that can be produced. Now the offspring could have different combinations of the dominant and recessive alleles. So here we need to do a cross between this big R allele and this little r allele in the gametes. Stick it together. That means we have big R, and our phenotype is smooth. Moving over here, our combination would be big R, little r, making our phenotype smooth, but instead of being homozygous, it's heterozygous. Here, we do a cross between little r and big R. Our phenotype, again, is smooth because big R, smooth phenotype, is dominant over little r, the wrinkled phenotype. So this genotype is also heterozygous. Now finally, we have the cross between the two gametes with the recessive allele for wrinkleness, little r and little r. This results in homozygous recessive genotype, and the phenotype is wrinkled. So here, taking a look at the zygotes that are produced out of everything, smooth peas outnumber wrinkled peas by about 3-to-1, because here we have half of our zygotes being represented by big R, little r, smooth phenotype, a fourth being represented by big R with a smooth phenotype, and a fourth being represented by little r with a wrinkled phenotype. So all of that to say, we have two very important terms. We have homozygous, meaning we have two cases of the same allele represented in the genotype, either two big R's or two little r's. And then we also have the case of heterozygous, where we have one copy of the dominant allele and one copy of the recessive allele. Sorry if that was extremely boring. I was just trying to go slow. So moving on. Understanding the mechanisms of evolution and how Mendelian genetics involves itself in population genetics. Genetic variation is the raw material of evolutionary change, so it's important to know how we can effectively measure that. And we should also ask the question, what are the factors that cause genetic changes within populations? That is, what mechanisms cause evolutionary change? In population genetics, evolution can be defined as change in gene frequencies through time. Population genetics tracks the fate across multiple generations of Mendelian genes in populations. Population genetics is concerned with whether a particular allele or genotype will become more or less common over time and what pressures are forcing that change. So let's talk about the intersection between what we know about Darwinian evolution by natural selection and what we just learned about Mendelian genetics. We know that survival and reproduction are not random. There has to be a correlation between fitness and phenotype, and that phenotype is driven by the genotype of an organism. So the factors that cause changes in the gene pool are these. And here I am defining gene pool as a sum of all of the alleles in a population. So here are several of the factors that change the gene pool composition in a population. Number one, we have selection, which is variation in fitness that is heritable, passed from parent to offspring. Next, we have mutation. So this is changes in DNA sequences. Maybe you have the emergence of a new characteristic or trait in your population. Number three is migration, the movement of genes across and between different populations. Next, we have recombination. This is the exchange of gene segments when we have sexual reproduction. Next, we have non-random mating, mating between neighbors rather than by just chance alone. And then finally, something called random genetic drift. And this primarily occurs in small populations, and sampling will result in different allele frequency from one generation to the next. We need to unpack this random genetic drift idea just a little bit in terms of the components that make it up, but I'm going to run through each of these before we get to that one. So number one, talking about selection. Selection occurs when a trait or a set of traits gives a survival advantage or a disadvantage. Selection in favor of a trait, that means that this trait helps individuals survive and live long enough to have viable offspring. We can also select against a trait, and that simply means that the trait is interfering with survival and reproduction. Traits increase in a population, typically, if they cause individuals to have more offspring. Traits decrease in a population if they kill off individuals before they can reproduce. So this is what we mean by selection. You can have a trait that is selected for or a trait that's selected against. Next, mutations. Small mutations can creep into populations over time. Some are helpful, some are harmful, and many are neutral, but may have a beneficial or harmful effect later. Mutations increase genetic variations in a population. We're gonna walk through one really exciting explicit example called tetrachromatic vision. This is a genetic mutation in the cones of our eyes. So in humans, the genes for cones are found on the X chromosome, the X sex chromosome. And women have 2 X's. Men only have 1, and that is why the incidence of color blindness is much more acute in men than in women, because if a woman has defective cone genes in one of the X chromosomes, the other one that's working better in the other X chromosome can just take over. Whereas if men have one defective cone gene in their X chromosome, they don't have another one to count on because the other chromosome is the Y chromosome. So tetrachromat mutants have 4 cones in their eyes. And this is in the purple, blue, green, red, plus yellow area. Most of the human population actually only has 3 cones in their eyes in the blue, purple, green, and red area. So an interesting test to see if you are a normal trichromat or a tetrachromat mutant is to take a look at this color spectrum. Okay. Now, I would suggest that you pause the lecture and count to see how many different colors you see and test yourself. If you can find between 20 and 32, you are a trichromat. That's fine, no shame. About 75% of humans are trichromats. But if you can see between 33 and 39 different colors, you might be a tetrachromat. And I could see about 39. So I might be a tetrachromat. I think that there are more detailed lab tests that you can take instead of just taking screen grabs from color spectra on the internet, but I think it's pretty exciting. Okay, migration. Migration can remove genes from a population, but it could also add genes to a population, or it could create a small population with different gene ratios than what was found in the founding population. And a good example of this is the founder effect that is observable today in Amish communities of Pennsylvania. There's a high incidence of polydactyly in these communities. That means that they have more than 5 fingers on their hands. You might be familiar with polydactyl cats. Cats often have polydactyly, and this is also a genetic thing, but that's when they have special thumbs, we have several community cats on my block with polydactyl limbs, and I call one of them Thumbs, and then I found another one and I didn't have a special name for him, so I just called him Orange Thumbs. So anyway, back to the Pennsylvania Amish. This community was founded by about 200 German immigrants, and so just a small sample of the German population came over to America. Now, whatever gene variants that they had in that small population were carried over here and then amplified. And so that's why we have a greater incidence of polydactyly among the Amish in Pennsylvania. This is also why we have a greater instance of recessive diseases in these small, tight-knit communities, because they're pretty insular because of cultural reasons. They do not have a lot of migration in and out of the population, and therefore the recessive alleles get amplified generation after generation. Sometimes that can have no effect at all, but sometimes it could be very deleterious. This is also why the trait for hemophilia is so rampant in royal families, for example, in Europe. Next, talking about sexual recombination or crossover events during sexual reproduction. These crossover events allow reshuffling of alleles during sexual reproduction, and it's a contributing mechanism that works with natural selection by creating combinations of genes that are selected for or against. So this is again contributing to more genetic variability that can then be expressed in the environment, interact with the environment, see if it works out, see if it should be selected for or against. And this is an image from your textbook. We have homologous chromosomes that are aligning during meiosis. We have chromosome crossover here, and then some of those spaces on the chromosome actually switch spaces. Next, we have non-random mating. Many living species have ways to compete for mates. This could be through actual physical combat or just singing prettier songs than your rival. In some populations, only a few individuals can mate because competition is that stiff. Mate preference is non-random. Even in the same species, many animals will prefer mates that resemble themselves. This also goes along with something called sexual selection. Sexual selection is the result of non-random mating, and this is when one of the two sexes, typically the female, selects for traits that she wants to have in her mate. Some are attractive and over the top, very colorful. The primary case is the selection of the peahen and her mate in the peacock. As you can see, it might be a drawback to select for something with such big feathery appendages that might make you especially slower in running away from predators, for example. But that is what over millennia the peahen has selected for. That is her expression of sexual selection. So through multiple generation after generation after generation of sexual selection, we have the grandiose display of the peacock and his feathers. Is that something that makes it harder for him to run away from predators? Sure, but it makes him more attractive for mating in the future with peahens. The entire conundrum of the peacock is something that really boggled the mind of Charles Darwin for a while. We can sort of explain it with something called the handicap hypothesis. I'm not going to talk about that here. That's something for, ecology lectures, but I just think it's super fascinating. You could have other drawbacks too, like, you got this great neck to fight off your potential competition, but then it makes it super, super hard for you to drink water at the watering hole. Next, we have random genetic drift. And this is just what it sounds like. It happens because of random chance. This primarily takes hold in smaller populations where you might have less genetic variability. And this ultimately culminates in the chance disappearance of particular genes because we have a random die-out population, or just randomly some organisms didn't get to reproduce in that generation. So after a while, the alleles that are passed down by the population might be more or less common than they originally were. Again, this is simply due to chance alone. And here we have an example from your textbook that illustrates with bunny rabbits the idea of random genetic drift. So in our first population, you'll see that we have the dominant B allele just as frequent as the recessive lowercase b allele. But due to random chance alone, only these 5 rabbits get to reproduce. After the next generation, we then see a shift in the frequency of those 2 different alleles, with the capital B being about 70% of the population and the lowercase b being 30%. The P and Q here we'll get to talk about soon, so don't worry about that. In the second generation here, instead of 5 rabbits being able to reproduce, only 2 get to reproduce. And this results in the entire eradication of that recessive lowercase b allele. And therefore, we've entirely lost the ability to have this albino white rabbit. So bouncing off of random genetic drift, we have 2 intertwined concepts. Number 1 is the bottleneck effect. This is the wiping out of a large portion of the gene pool. This is typically associated with natural disasters that just kill randomly. And then we have the founder effect. This is a small group of individuals that are founding a new population, reducing the gene pool, and only bringing with it the genes that it had already. This is a pretty standard illustration of what we mean by the bottleneck effect. This represents a drastic change in population size, thereby decreasing the genetic diversity in later generations. So this is a sudden wiping out of the entire diversity of the gene pool. We don't have, everything eradicated at once, but here the survivor's genetic structure becomes the entire population's genetic structure. And that could be very, very different from the pre-disaster population. So here we have a bottle representing with different colored candies the different gene variants in the population's gene pool. We see that we have equal numbers of red, blue, yellow, and green alleles, okay? The bottleneck, here where the bottle actually tapers at the neck, this bottleneck event drastically reduces the size of the population. So our event is pouring that bottle into a little jar and seeing what comes out, okay? By chance, the gene pool of the reduced population in the jar is actually mostly blue with a few yellow alleles in there, okay? And so after generation of only having reproduction events in this smaller population, our entire population then is going to be represented by blue and yellow alleles. We have completely eradicated the diversity of other colors. We no longer have red and we no longer have green, okay? This reduced genetic variation results in low adaptability. It means that the population may not be able to adapt to new selective pressures like climate shifts or a shift in available resources, because the genetic variation that selection would act on has drifted out of the population. We no longer have the genetic variation in our gene pool to allow for adaptation and natural selection. Coupled to this is something called the founder effect. This is the establishment of a population by a few individuals, and this can profoundly affect genetic variation in subsequent generations. The consequences include fewer alleles, fixed alleles, and modified allele frequencies compared to the source population, perhaps due to a new environment. And here's a little schematic representation of what that looks like. Boop. Okay, so we should watch that again, but slower. So for the founder effect, we reduce our genetic variation by sending across the river here only one white mouse. Okay, the rest of the population are black, and therefore the generations over here that are to come are going to be representing more of that black phenotype than the white phenotype, even though in the original population they were equally divided. And now with the bottleneck effect, we have this random natural disaster, which is this lightning bolt coming and eradicating half of the population. Poor mice. But again, the surviving population is largely dominated by the black phenotype. So subsequent generations, therefore, are not going to have white represented nearly as much as the black phenotype. We have explicit examples of the bottleneck effect that we can observe today. So here is a northern elephant seal. They have reduced genetic variation because of a population bottleneck that was inflicted upon them in the 1890s. There was excessive harvesting of the northern elephant seal because they produce lots and lots of oil. And so people were harvesting them for their oil so much to an extent that we reduced their population size to as few as 20 individuals at the end of the 19th century. What's interesting is that since that time, their population has actually rebounded. There's over 30,000 individuals of the northern elephant seal in their population. But when you take a look at their genes and their genetic variation, they still have evidence of that bottleneck. They have much less genetic variation than a population of southern elephant seal that wasn't nearly hunted as much. So even though in numbers, it seems that they are doing okay, they still don't have the genetic variation and a diversity that would allow them to successfully adapt to their changing environment in the future. We also have historical examples of bottlenecks, particularly with the potato blight that devastated the Irish community in 1845 to 1849. This is because there was reduced genetic variation in the potatoes that were used in Ireland. And this is actually one of the greatest factors that led to mass immigration from Ireland to the United States during this time. All potatoes in Ireland were introduced at once from one population of potato in South America. Okay, this in itself was a genetic bottleneck. What makes it worse is that these potatoes were not grown agriculturally through seeds and plants that could potentially introduce mutations and genetic variation. Instead, they were grown and propagated through tubers, which are clones from the mother. Okay, and this again reduced the chance for genetic variability. And all the alleles in the population, therefore, were the same, making them susceptible and vulnerable, all in the same respect to whatever disease that might hit it. There was not enough genetic variation to have many survivors. So nearly all of the potatoes in the country were killed because of this potato blight that was brought over from Mexico. So concluding story about all of this is that population size matters, and it matters a whole lot. These random events will have a larger effect on a smaller population size. So taking a look at this chart here, we have generation going from generation 0 to the left and then all the way to generation 6 and a hypothetical population on the right. And then we have frequency of a dominant allele here. If our population size is 10,000 individuals, the allele frequencies are going to remain constant, okay, no matter where we look across different populations. But if we go with a population of size 4 individuals, that small population means that one allele may become extinct within just a few generations. You'll have rapid, drastic changes just because the population size is so extremely small. All of this is explained wonderfully in a video on TED Ed that I found. This is from a group called the Amoeba Sisters, and they have a bunch of lovely, sweet videos. So I really hope that there's a lot more material that they cover that I can incorporate in what we're doing. So we need to ask the question, what does a population at equilibrium look like? We talked about all the things that spur about change, but what about a population at equilibrium? And I must warn you, this is where the math comes in. I know a lot of us are excited about biology because it doesn't include a lot of math, but I hate to tell you that, yeah, we need to get into some math. And it's not so bad, it's really not. Particularly, we're talking about Hardy Weinberg equilibrium. This may be something that you've heard about before. If not, we're gonna walk through some examples in the next video using the expertise of Khan Academy. But the Hardy Weinberg equilibrium model is a null model. And as we said before, all models are wrong, but some are useful. And this is useful because it allows us to predict genotype frequencies within a population really, really well. And this is the null model, or the null hypothesis, is that the population is in Hardy Weinberg equilibrium. This could be any old population. The Hardy Weinberg equilibrium calculation can be applied to all different types of populations. However, there are assumptions. As with any model, there are assumptions that go into it. So we are, number one, assuming that we are dealing with a diploid autosomal locus with two alleles, represented with a big A here and a little a here. Diploid means there are two copies of the chromosome. Autosomal means that it's not a sexual cell, it's a somatic cell, it's a body cell. And locus means the location on the genome. And two alleles means that there's two alternatives, either the dominant or the recessive allele. Number two, we're dealing with a sexual organism. Okay, so not all organisms are sexual. There are some asexual things. So what I said before about this applying to all populations, okay, there are some restrictions. We have this basic schema here where the parents are diploid, meaning we have two copies of the genome. They produce individually their own gametes, which are haploid, containing one copy of the genome. They then pair up to form the zygote, which is a diploid organism, again, containing two copies. The gene pool contains two alleles, big A and little a. We have more assumptions. Here we go. Number three, that we have a large gene pool. Remember what I said about population sizes? We needed to have a big population size in order to accommodate the Hardy Weinberg model. 4 generations are non-overlapping, meaning we don't have the F1 generation mating with someone from the F2 generation or the F1 generation mating with their parent. That is not happening. Number 5, we have random mating. So no sexual selection. Number 6, no migration. We're not moving genes in and out of a single population. No mutation. There's no new emergence of characteristics or genes. And then finally, no selection on genes. Okay, so in the real world, this is not happening. There's no population for which natural selection is not a factor. But as we said before, this is a useful model for making predictions about what evolutionary forces are acting on a population to kick it out of equilibrium. So here's where the math comes in. Brace yourself. We can approximate gamete or allele frequencies in this way. The frequency of A, the dominant allele, is represented by the letter P. The frequency of the recessive allele, lowercase a, is represented by the letter Q. In any population, P plus Q equals 1. Okay, seems pretty fair enough. If there are only 2 alleles for a trait in a population, then the frequency of the dominant allele plus the frequency of the recessive allele is going to equal 1. Here in our example, Purple is dominant to pink. Okay, so this is just talking about frequency of dominant and recessive. But remember, genotype and phenotype is the expression of two alleles together. This is where it gets a little bit more complicated. Genotype frequency of 3 possible genotypes, okay? We can have homozygous dominant genotype, which is depicted as big A, big A. We can also have heterozygous dominant, which is represented by big A, little a. That big A is dominant over the recessive little a. And then finally, we have homozygous recessive, 2 little a's. All of this works out to this, with this math. The frequency of our homozygous dominant is P sub A times P sub A equals P sub A squared, okay? We can do that same type of equation for heterozygous dominant, and then homozygous recessive. We can sum these terms up together and have it all represented in this equation here. P squared plus 2PQ plus Q squared equals 1. So if there are 2 alleles for a trait in a population, then the frequency of homozygous dominant genotype squared plus 2 times the frequency of heterozygous genotype plus the frequency of homozygous recessive genotype squared equals 1. The main points of all of this is that P plus Q equals 1. So more generally, the sum of the allele frequencies equals 1. P squared plus 2PQ plus Q squared equals 1. In other words, the sum of the genotype frequencies equals 1. So here we're dealing with allele frequencies. Here we're dealing with genotype frequencies. Okay, make sure you remember and understand the differences and the distinctiveness between the allele frequency and the genotype frequency. And 2 populations with markedly different genotype frequencies can have the same allele frequencies. So how do we do the calculations? It's actually not that difficult. I want to leave it to the experts at Khan Academy to explain this to you. They do a really good job in the Hardy Weinberg population genetics series. I'm going to post 4 Khan Academy videos that walk you through allele frequencies, Hardy Weinberg calculations, and then the implications of the Hardy Weinberg equilibrium on the tartan. So here on the tartan, I've strung together the 4 Khan Academy videos for you. They're about 30 minutes. And then I've also posted them in this PowerPoint. So this PowerPoint called Calculating Hardy Weinberg Equilibrium just contains those videos that are already posted on the tartan. So no need to download this if you viewed the videos online. Part of your pre-lab this week is going to be going through the Khan Academy activities, starting from allele frequency and the gene pool, all the way through practicing selection and genetic drift. Our activity in class is going to be putting Hardy Weinberg equilibrium in practice and tracking the change in frequency of different alleles. So be ready to come to class knowing how to calculate Hardy Weinberg and what allele frequencies mean.